I have been a beekeeper for 3.5 years now. I joined the Honeybee Club my first year at UF, and I've always loved bees ever since sixth grade. So, for my senior thesis, I decided to do my project analyzing honeybee data from the United States Department of Agriculture. They have a National Agricultural Statistics Service. It's a mouthful. But anyways, everybody should know what a honeybee is, but they play a very important role in agriculture and our ecosystem with their very important pollination services. But in the early 2000s, beekeepers started reporting alarming losses. So my project basically did three things. It looked at historical trends based on the data that I was given. I looked at the factors that are associated with colony health, such as pests, pesticides, the varroa mite. If you come to talk to me, I'll talk to you about varroa. And then also I developed some predictive models to see where the trends would go in the future over the next 10 years. And yeah, so hopefully you buzz on by.
Okay, thank you, Mallory. So for the next person, oh, wrong way. We have Enrique. So if we get a round of applause for Enrique here.
Hello, everyone. My name is Enrique Medici, and I'm a first-year PhD nuclear engineering student. And my project deals with machine learning for fundamental spectroscopic constants. And essentially, where we have optical emission spectroscopy serving as a tool for diagnostics of reactive flows of shock tubes, elemental abundance in stars, and characterization of laser-produced plasmas, where that's the focus of this work. And by collecting light, we obtain spectra with peaks that are unique to the emitter, and we can readily identify these peaks. But to gain more information regarding the system characteristics, spectroscopic coefficients must be known. And specifically, the Einstein coefficient is directly related to the intensity of these peaks, and it is crucial to estimate plasma temperatures. For materials that are specific for nuclear forensics, there is no sustainable approach to obtain these values, and therefore we applied machine learning to develop a generalized solution to this problem. We were able to outperform the baseline in both data and performance scopes, and we applied the model to generate a temperature curve for our plutonium plasma, which was previously unknown. Thank you.
Okay, thank you, Enrique. And the next person up is whose ever slide this is. Let's go give a big round of applause.
Hey everybody, my name is Alexander Mikienko, and I am a member of the Quantum Information for Entanglement and Enhanced Sensing Research Group over in the Physics Department. We seek to utilize the fundamental properties of quantum mechanics to enhance the sensitivity of measurements that can be made in a lab. Specifically, my research deals with the implementation and design of a control system to control molecular ensembles for such quantum metrology. Specifically, our experiment aims to create spin squeezing in strontium fluoride molecules to surpass what is known as the standard quantum limit, which is a technical limit to the sensitivity achievable with non-interacting particles. This experiment requires a time resolution on the order of microseconds, for which a hardware timable, synchronized, and deterministic control system is necessary. Indeed, we have verified the synchronization of our control system to 8 nanoseconds, which corresponds to a phase uncertainty of roughly 100 or 1,000 times smaller than the standard quantum limit, thus allowing us to probe in that regime. Pushing the sensitivity of molecular ensembles beyond the standard quantum limit opens up pathways to potentially exploring and detecting physics beyond the standard model, including the search for symmetry violating dipole moments and dark matter detection. Thank you.
Thank you so much. Next person we have is whoever slide this is. No, you're good. It looks really good.
Hey everyone. Today I'm going to be introducing a deep learning pipeline to essentially convert like a protein and basically optimize it to increase its binding to a sugar. And basically the way this is done is through a whole pipeline starting with ligand MPNN. And basically it's a 3-block neural network program that could actually learn from the structural information from the protein and the sugar and essentially autoregressively decode a bunch of sequences that are covered up here. And it can make thousands of these candidates that are supposed to be better at binding to the sugar. And then basically through a competitive binding assay with AlphaFold3, it essentially models whether the whether the sugar goes to the new optimized protein or the old original one. So if it goes to the new one, that means it's better. And basically you could use this to narrow down the 3,000 candidates or however many you make into one optimal sequence. And that could be used for whatever you want. So for this project, I use it for optimizing universal blood type conversion. So basically cleaving the sugars off of red blood cells for type A blood and turning it to type O blood. But this could be used for any metabolic pathway in human health, and it could definitely save thousands of lives. Thank you.
Thank you. That was amazing. For the next person, we have Theo.
Hello, everyone. So I assume all of us have used a computer here. But what if you didn't have the ability to use a mouse? So a mouse is very important to use a computer, and 7% of US working adults can't use a mouse or struggle to use a mouse, and that's a huge number of people. So there have been many attempts to solve this problem and assist these users in using a computer, including like voice-based navigation or eye tracking. And the problem with eye tracking is that small precision errors can make it impossible to like reliably target small elements. So recently, with vision language models, which are just LLMs that can take both image and text as input, a lot of researchers have tried to have the vision language model get X and Y coordinates of a target element. So you give it a screenshot like this and say, click the Finder icon, and then it will basically find the coordinates of the Finder icon, and then with the Python script, you can click it. So the main problem with that being that it takes too long to run that inference for it to be useful. So what we do is we use the eye tracker reading to kind of guide the VLM to focus on the right region, and that drastically reduces the inference time and makes it realistic for deployment even on edge devices. So you don't even need the cloud. Everything can run on device in under a second on a MacBook Pro M1. So if you're interested to learn more, have a look at my poster.
Theo. For the next person, we have, I just went back. This person right here, Alex.
Hi, everyone. So we all know that we have a genome in all of our cells, and like it's about 1.8 centimeters long if you stretch it out all the way. But obviously, it has to fit in a small nuclei of about like a few micrometers in diameter. So the question is, it has to go undergo all this folding. What's the significance of this folding? So one of the big problems in genomics right now is the problem of structural variation, which is like alterations in our genome. And a lot of the times we know what it's doing, like it's deleting a whole gene, for example. Like a whole gene gets deleted, we know that protein's not going to get expressed. But sometimes we have no idea what it's doing, and that's partly because we're not looking at the 3D impacts. So basically, we created a model to look specifically at the 3D impacts, like these organization elements, and we found that our model, when we included the 3D impacts, performed much significantly better than when we did not include them. All right, thank you.
Thank you, Alex. For the next person, we have Price. So you can round of applause for Price.
Hi everyone. I'm Price. I'm a junior computer science student, and my project is on fairness-aware AI. This study examines how transcription fidelity impacts language model sentiment inference in oral history interviews with older African American English speakers, and in the future will include older speakers of other non-standard American English dialects. We compare real human-written transcripts of these interviews with hand-corrected phoneme-level drafts from a speech-to-text model, isolating differences between them, such as omitted pauses, discourse particles, and hesitation markings. Sentiment predictions for these transcription types were evaluated across eight small and large language models in both zero and few-shot testing environments to provide a representative cross-section of modern language models of different sizes, opacities, and distribution policies. While these transcription differences were rarely found to be the outright cause of sentiment label reversals, they introduced subtle, model-specific sentiment drift and sentence-level instability, zero-shot configurations exemplifying systematic bias against older speakers, while few-shot testing minimized it. And the omission of hesitation and discourse markers had the greatest impact on sentiment inference. Future work will surround the application of this methodology to more non-standard English dialects and the introduction of topical analysis to help explain systematic sentiment drift. Thank you.
And the next person is Braden. So give a round of applause for Braden.
Hello, good morning, everybody. I'm Braden. I'm a third-year computer engineering student, and my project is a new approach to searching high-dimensional data with a semantic structure pretty efficiently. Currently, current ANN search techniques rely heavily on geometric similarity, stuff like k-nearest neighbors. But the issue arises with a lot of practical data in the real world has some sort of semantic structure. So our approach is what we call the PHBF, which combines a directed acyclic graph alongside a counting Bloom filter with locality-sensitive hashing. This allows us to make search requests that can be like, give me this or this that relates to the semantic structure. Lots of complex Boolean queries, etc. Our results are very promising and very exciting. So if you're interested at all, please come see me. I'd be happy to tell more. Thank you.
Thank you, Brayden. And the next person is Ethan.
Morning, y'all. So target stance extraction is the task of taking some sort of document, normally a social media post from X or Facebook, and predicting both a target for that document, what it's talking about, and its stance, its opinion toward that target. So the original target stance extraction system used a two model neural pipeline to predict that. So you have one neural model, makes a pass over the document, predicts a target. That gets pipelined into a second neural model, looks over the document again, and has that target as context, predicts the final stance. We're able to compress that into one model pipeline that makes only one pass over a document, predicts both of those jointly. This reduces the model parameter footprint by about 50%. It also makes these systems much, much easier to train and deploy. So whether you're trying to track public opinion on a new consumer product, a new political candidate, or something more fun and mundane like The Bachelorette, we've made it far more easy for you.
Thank you, Ethan. And the next person is Sakith and Anna, Omar, or Gabriel. See, it's Anna. Round of applause.
Hi guys, I'm Gabriel. I'm a fourth year statistics student.
And my name is Anna, and I'm a third year stats major. And we're presenting boosting reading fluency with interactive digital blending drills. So blending drills are exercises that assist with decoding in younger students. What decoding is a skill that helps students turn written words into like a sounded form. And it's actually one of the most crucial skills that students learn before they reach reading fluency. And blending drills are an exercise, like I said, to practice decoding. And what it does, basically, it changes each individual letter one by one, where students sorry.
Yeah, so we have two different ways of implementing these blending drills. One is a traditional way with an algorithm, and then the second way is calling a large language model. So we essentially have a bag of words, and these are provided through the College of Education, and we run them through whichever model we use, and then we run an evaluation script on it to see if like the words are good enough for the students. And then these are some graphs, and when we finish, we're going to implement it with this Stories app with the virtual learning lab. And
yeah, our goal is basically to create a full stack gamified system for students to learn this, which could actually also potentially help with analyzing reading difficulties, which is really crucial. Catch that early on rather than later.